Hello, and welcome to the second lecture. This will be Myocyte Molecular Physiology. Another name for this lecture could have been Cardiac Cell Biology or Myocyte Microanatomy and Physiology, just to give you an idea of some of the topics we'll be talking about. We're going to be starting right where we left off in our first lecture. We were talking about the cell membrane and electrical chemical physiology and the action potentials that occur around it. And we ended when we zoomed out from one cell to multiple cells and started talking about how these action potentials were conducted from one cell to another. And that's where we're going to start with our second lecture here. We will be looking at the anatomy of the myocyte and doing it in a functional approach. And we'll start off by looking at how these cells are connected, and we will progressively zoom in into further and further subunits. And once we reach our basic functional unit, we'll start talking about the molecular physiology of how the cell works. We'll do this for both the cardiac cell and the muscular cell within your vasculature as well. The second half of this lecture in particular lends itself very well to a dynamic visual teaching medium, so we'll be spending a lot of time in the classroom. But for now, let's head to the lecture hall and start myocyte molecular physiology. Myocyte molecular physiology. We'll start off talking about the cardiac myocyte itself. Cardiac myocytes are types of striated muscle. They are similar to skeletal muscle. Generally, their size is about 25 nanometers in diameter and 100 nanometers in length. Ventricular myocytes are larger than atrial myocytes. However, both ventricular and atrial myocytes are shorter than normal skeletal muscle. Cardiac myocytes are single nucleated, and they are aligned end to end when they connect with each other. They do have branch points, however, these branch points as well lined up end to end with adjacent cells. This organization of cells forms what is called a functional syncytism. The terminal portion of the myocyte forms an interdigitated region, zigzag in appearance, called intercalated disc. These discs are important because they serve as insertion points for the myofibrils, the myofibrils being the contractile unit of the myocyte. Desmosome structures are firm intermittent attachments located between the membranes and are formed by adherent proteins. They form intracellular bridges and add structural integrity to the functional syncytism. Desmosome structures are not associated with myofibrils. Gap junctions are low-resistance electrical connections formed by connexin proteins in the form of six-membered rings on each end of the cell membrane. Connecting, this allows communication between sarcoplasmic intracellular spaces, thus permitting cell-to-cell ion current and conduction, the main topic of the final portion of our last lecture. Going through the process of progressively zooming in on the myocyte, when we look at the grouping of cells, the functional syncytism, one of these cells is our cardiac myocyte, the sarcolemma, and within the cell there are myofibril bundles. These are bundles of myofilaments, usually about 300 myofilaments per bundle, and each myofilament is made up of sarcomeres, and the sarcomere is the basic functional unit of contraction. Your sarcomere generally is 1.6 nanometers in diameter to 2.2 nanometers in length. It is important to note that the length of the sarcomere determines the force of myocardial contraction, and this is a concept we'll come back to at the end of the lecture. Sarcomeres are made up of numerous structures. The two most important are filaments, and your two filaments are your thick filaments, which is myosin, and your thin filament, which is actin. And these filaments are arranged in a hexagonal arrangement such that for every thick filament, there are 6 thin filaments surrounding it. When we look at figures and graphs throughout this talk, we'll be seeing two-dimensional representations of myosin and actin in a one-to-one relationship. And what you need to keep in mind is that in reality, there are 6 thin filaments for each myosin, and multiple of these thin filaments are participating with multiple myosin in this three-dimensional structure. Thick filaments, or myosin, is made of 2 heavy chains which are intertwined helical tails, and they are positioned end to end, and their meeting point is what is called the M-line histologically. It is the area of myosin tail connection. On the distal ends, there are 2 globular heads. They are attached to the heavy chains by hinge molecules. On these globules, there is a site of ATP binding and myosin ATPase, which is an enzyme that hydrolyzes ATP. The globular head interacts with the binding site on actin, forming the actin-myosin cross-bridge. ATP is required for the cross-bridge formation and the linkage between thin and thick filaments. Thin filaments are composed of actin. Actin is a repeating globular monomeric protein unit called G-actin, and when this G-actin is arranged in polymerized filaments, it is called F-actin. Two F-actins are arranged into a helical structure. Actin binds to the myosin globular head, forming the actin-myosin bridge complex, and it activates the myosin ATPase, which hydrolyzes ATP to ADP and inorganic phosphate. Tropomyosin, or TM, is a rod-shaped, lightly coiled protein chain. It is interdigitated between the actin strands. So the two actin polymerized helical filaments form a groove, and tropomyosin lies within that groove. Each tropomyosin is associated with 7 actin molecules. Tropomyosin is bound to regulatory proteins called troponins. Troponin, or TN, are regulatory complexes which are attached to tropomyosin at regular intervals. There are 3 subtypes: troponin T, troponin C, and troponin I. Troponin T is structural in function only. It binds the rest of the troponin complex to the tropomyosin unit. The T in troponin T stands for tropomyosin. Troponin C, and the C in troponin C stands for calcium, is the binding site for calcium ions during the excitation-contraction coupling process. Excitation-contraction coupling is the process of changing an electrical action potential, discussed in the first lecture, into a contractile process, which will be discussed in this lecture. When bound to calcium, troponin C removes the inhibitory effects of troponin I. Calcium binds in a concentration-dependent manner to troponin C as long as cytosolic calcium concentrations remain high. This binding causes a conformational change, moving the troponin-tropomyosin complex away from the myosin binding site on actin and exposing it. This makes actin accessible for the myosin globular head. When calcium is removed from troponin C, the troponin-tropomyosin resumes its inactivating position. Troponin I, where the I stands for inhibitory, troponin I is the inhibitory protein which inhibits the myosin binding site on actin. Tintin is a large filamentous protein that connects myosin to the Z lines. The elastic properties of tintin contribute to the passive mechanical properties of the heart. So we'll do a quick summary of our filaments. Again, with our thick filament, which is myosin, we're going to have a heavy chain and 2 globular heads associated with light chain heads. And you're going to have 2 of these molecules positioned tail to tail, such that they look like this. Now, your thin filament, or your actin, is going to be made of G-actin, which is a globular protein. And when these are arranged into a long filament, they become F-actin, and 2 of these are going to be positioned in a helical structure. And lying in the groove that this helical structure makes is tropomyosin, and binding to tropomyosin is your troponin complexes. We have troponin T, which is functional and structural, just binds to tropomyosin. We have troponin C, which binds to calcium, and then troponin I, which blocks the binding site for this myosin globular head. And when calcium comes along and binds to troponin C, the position of your troponin I changes. It moves out of the way such that this binding site on actin is available. Having gone through the basic functional microanatomy, we'll take a look at the sarcomere histologic nomenclature so we can build a language and describe the changes that occur during contraction. Within your sarcomere, you have lines, bands, and zones. Your lines describe transverse structures within the sarcomere, and your bands describe longitudinal structures within your sarcomere. They run along the distance of the sarcomere rather than crossing it like the lines. You have 2 lines. You have a Z line and an M line. Your Z line is the transverse septum matrix, and it is made of alpha actinin. So it is made out of the same material as the thin filaments, and the thin filaments are connected to this landmark. The Z-line to Z-line space, or the inter-Z-line space, denotes the boundaries of the sarcomere and describes it as a functional unit. Your M-line is the nodular thickening of the thick myofilament. It is the binding site of the two heavy chain tails of myosin. It denotes the center of the H zone. You have two bands. You have an I band and an A band. Your I band is the longitudinal space between thick filaments, or myosin filaments. The Z line is the middle of your I band. Your A band is the length of the thick filament, or the length of the two myosin complexes. The M line is the middle of the A band. Your zone describes an empty space. Your H zone is the space between thin filaments. Sliding filament theory is the theory used to describe the process of contraction of your sarcomere complex. Actin and myosin slide past each other during excitation-contraction coupling. This is accomplished by a ratcheting movement between the myosin heads and actin. The distance between Z-lines, or the length of an individual sarcomere, shortens as this contraction takes place. And let's take a more in-depth look at the actin-myosin binding mechanism. The first thing that needs to happen is that ATP needs to bind to the myosin globular head. A conformational change in the myosin globular head is brought about when ATP is hydrolyzed to ADP and inorganic phosphate. At the same time, calcium is binding to troponin C and inhibiting the inhibition of actin by troponin I. This allows for an actin-myosin bridge to form. The myosin globular head then moves with its hinge-like movement back to its original resting position with the release of ADP and inorganic phosphate. This hinge movement brings about the sliding of actin and myosin past each other, and thus sarcomere and subsequent muscle contraction. Conformational change of the myosin head back to a potential state of energy comes with the next ATP binding of myosin, and calcium is removed from troponin C, and troponin I re-inhibits the actin binding site until calcium then rebinds to troponin C again. This process is repeated over and over until the concentration of calcium is decreased. Let's review the mechanism of actin-myosin cross-bridging. If we have our myosin molecule here with the myosin head in its resting state, and we have our F-actinin and our alpha-actinin, which is our Z-line, the first thing that we're going to need is for ATP to come in and bind to the myosin head, and at the same time calcium is going to come in and bind to troponin C, removing the inhibitory effect of troponin I, exposing the binding site on actin for myosin to bind to. You can see that there's a conformational change within the position of the myosin head, and right now ATP is bound to this, when it will be hydrolyzed to ADP and inorganic phosphate. And when this releases, you're going to have a change in the position of the myosin head such that it will bend back to its resting state position. And when it does so, it will move your actin filament one way, your myosin filament the other, and the filaments will slide past each other, and you'll have contraction. ATP then needs to come and rebind for removal, and calcium concentrations need to lower, in which case you'd end up back in your resting state. This process will continue until calcium concentrations are decreased enough such that the binding site is no longer present. Keep in mind, even though this is a one-to-one drawing and it's a two-dimensional representation, for each myosin surrounding it, you have 6 actin. So it's really happening in a circle and three-dimensionally. And it's also happening in multiple places. There are multiple myosin heads, such that you get this sort of ratcheting phenomenon as they slide past, as the actin and myosin slide past each other. The histologic changes and consistencies that we see during contraction are that your A band is consistent in length. And remember, your A band is your myosin or your thick filament length. So your myosin does not change in length. Your I band, which is the space between myosins, does shorten, as does your H zone, which is the space between thin filaments or actin. So the structures themselves don't change, only the relative spaces between the structures change. We'll do a quick visual summary of our sarcomere histologic nomenclature again. We have our thin filaments right over here, and this is our G actin, which when in a chain form F actin, and they are connected to your alpha actin, which form your Z lines. You use Z lines to find the boundary of your sarcomere. Within this histologic nomenclature, lines denote structures that cross transversely across the sarcomere, and bands denote structures that run longitudinally. So the first line that we're talking about is our Z line or alpha actin in the end of the sarcomere, connecting our thin filaments. Okay? The next line that we'll talk about is our M line, and it is the meeting head of the two tails of our myosin, our heavy chain. So remember, we have our helix, one on each side, globular heads, light chains associated with it. And where these two heavy tails meet, there is a thickening, and this is your M line. Think M for myosin. And so these are your two transverse lines that cross your sarcomere. Your bands move longitudinally. Your first band is going to be your A band. And your A band is the distance of your thick filament or your myosin. Your I band is going to be the distance between thick filaments, or myosin head to myosin head. So the center of your I band is your Z line. The center of your A band is your M line. The empty space is a zone, and this is your H zone. It is the space between thin filaments. So when you contract, the length of your myosin or your A band doesn't change, and the length of your thin filaments, which actually isn't measured by anything in this nomenclature, does not change either. What changes is your H zone. The two get brought closer together. H zone gets smaller, and your I band, the space between your myosin thick filaments, gets shorter as well. At this point, we've now discussed the basic functional contractile unit of the myocyte, and we need to take a look at the rest of the cellular machinery, which will be responsible for activating this functional unit. Your sarcolemma membrane of the myocyte surrounds the bundles of myofibrils, and within this membrane there are very deep invaginations which increase the surface area of the membrane and expose more of the cell and more of the intracellular portions of the cell to the extracellular environment. And these deep invaginations are called tubules. You have two types of tubules. You have transverse tubules, which move perpendicular to the length of the sarcomere and are at the outer portions of the cell. Deep within the cell, you also have longitudinal tubules, which run along the length of the sarcomeres. Again, the purpose of the tubules is to increase the organelle membrane proximity and to maximize surface area for electrolyte exchange. Adjacent to these cell membranes in these tubules is your sarcoplasmic reticulum. It is in close association with the T-tubules and the surrounding filaments. The main function of your sarcoplasmic reticulum is to regulate intracellular calcium concentrations. Calcium inside of the sarcoplasmic reticulum binds to the most common storage protein, calsequestrin, and there it is held in the cisterna terminala until it is released. A significant number of mitochondria surround the sarcoplasmic reticulum to supply it with ATP. SERCA is sarcoplasmic reticulum calcium ATPase. It is a sarcoplasmic reticulum calcium ATPase dependent calcium pump. located on your sarcoplasmic reticulum. We also need to remember our calcium cell membrane machinery that we discussed in our last lecture. We have our slow inward calcium channels, sodium-calcium exchangers, and calcium ATP pumps. The terminal cisternae is a sarcoplasmic reticulum end pouch connecting structure between the sarcoplasmic reticulum and the T tubules. It is composed of two substructures, your RYR and your feet. Your feet are sarcoplasmic reticulum projections. These are dense strands which connect the sarcoplasmic reticulum to the cell membrane. The purpose is for them to sense calcium between the T tubules and the terminal cisternae. This is a very electron-dense region. Your RYR is your ryanodine receptor. It is a calcium release channel. Again, abundant mitochondria surround the sarcoplasmic reticulum and are in close association with it and provide the large ATP energy source needed for its performance. So at this point, having gone through the discussion of functional microanatomy of your myocyte, we're at the point where we can now build a bridge between the first lecture, which focused on excitation, and this lecture, which will focus on contraction. So excitation-contraction coupling is the coupling between the myocyte's action potential and the myocyte's contraction. Calcium enters the cell through slow inward L-type calcium channels during depolarization. The calcium that enters the cell is called trigger calcium. It's actually a very small amount of calcium. The calcium that enters during an action potential is not sufficient enough to generate contraction alone. So there is the concept of calcium-induced calcium release. What we will see is that the amount of calcium entering the cell with the action potential is very small in comparison to what will be released by the sarcoplasmic reticulum. This trigger calcium is sensed by the feet of the RYR of the terminal cisterna in the electron-dense region that is there. Calcium then triggers the release of more calcium from the terminal cisterna stores, and the intracellular calcium is increased 100-fold. It is this calcium, the calcium that is released from the sarcoplasmic reticulum, that binds to troponin C in a concentration-dependent manner, bringing about the conformational change within the troponin complex. Myosin heads then have the ability to bind to actin, creating the cross bridge and subsequent movements with ATP hydrolysis. Sarcomere length is decreased by the ratcheting movement of our sliding filament theory. Calcium is eventually then resequestered into the sarcoplasmic reticulum by the Sarca ATP pump. Calcium is removed from troponin C as intracellular calcium levels decrease, causing myosin to unbind from actin, a process which also requires ATP, and sarcomere length then increases. Now that we've completed examining the cellular machinery and its function, we can build towards larger concepts. And the two we'll be dealing with are inotropy and lusitropy, which is the contraction and relaxation of the heart, both of which are energy-dependent processes. We're going to be looking at inotropy first and inotropy regulation. Looking at our cellular machinery, you'll find that there are two main substrates needed for inotropy to take place, and that is calcium and ATP. Inotropy can be regulated through the modulation of these two substrate parameters. Most of the methods that you will see for regulating inotropy have to do with calcium modulation. Only one of them has to do with ATP modulation. The way that we can affect calcium within the cell are as follows. First of all, we can affect the calcium entry into the cell through the slow inward L-type calcium channels. We can affect calcium release by the sarcoplasmic reticulum. We can affect calcium binding to troponin C. And we can affect calcium uptake into the sarcoplasmic reticulum by the Sarca activity and calcium efflux across the sarcolemma and out of the cell. The ATP method of regulating inotropy focuses on myosin phosphorylation. So going through each of these mechanisms individually and in detail, first we'll deal with the slow inward L-type calcium channels and trigger calcium entry into the cell. Sympathetic nerves will release norepinephrine locally, and your adrenal glands will release epinephrine and norepinephrine into your circulation systemically. This epinephrine and norepinephrine bind to your beta-1 adrenoreceptors. Your beta-1 adrenoreceptor is coupled with a stimulatory GS protein, a specific guanine nucleotide binding regulatory protein. This GS protein activates adenyl cyclase, and adenyl cyclase is responsible for the hydrolysis of ATP to cyclic AMP. Cyclic adenosine monophosphate is a secondary messenger to activate protein kinase A. Protein kinase A phosphorylates the L-type slow inward calcium channels and increases their permeability to calcium during the action potential. Thus, you have increased trigger calcium. To summarize the first method of inotropy regulation, if we have epinephrine or norepinephrine, these will bind to their receptor, beta-1 receptor. Beta-1 receptor is coupled with a G stimulatory protein. This is going to upregulate our adenyl cyclase. Adenyl cyclase, in the presence of ATP, is going to make cyclic AMP, and cyclic AMP is going to activate protein kinase A, and protein kinase A will phosphorylate our calcium slow inward L-type channel, thereby increasing the influx of our trigger calcium and increasing our inotropy. The effect of calcium entry into the cell by L-type channels can be inhibited as well as stimulated. In the case of vagal nerve release of acetylcholine locally, acetylcholine will bind to muscarinic or M2 receptors. These muscarinic receptors are coupled to inhibitory GI proteins, and these GI proteins inhibit adenyl cyclase. Once inhibited, adenyl cyclase cannot hydrolyze ATP to cyclic AMP, and the further molecular physiology is inhibited. The end result being that there is less permeability of calcium through the L-type calcium channels during an action potential. To summarize inhibition of this pathway, if we have acetylcholine or adenosine, these will bind to their receptors, or muscarinic receptors. This will be coupled to a G inhibitory protein, which is going to have a negative effect on adenyl cyclase. Therefore, our ATP will not convert to cyclic AMP, and the rest of the pathway will not be carried out. We will not have pKa and phosphorylation of our slow inward calcium type channels, and we will not have an increase in trigger calcium. There is a major and minor pathway through which calcium release from the sarcoplasmic reticulum is modulated. Sympathetic nerve release of norepinephrine locally, or again, adrenal release of epinephrine or norepinephrine into the systemic circulation will make epinephrine and norepinephrine available to bind to beta-1 adrenoreceptors. Beta-1 adrenoreceptors are coupled to, again, stimulatory GS proteins, specific guanine nucleotide binding regulatory proteins, and these GS proteins activate adenyl cyclase. Adenyl cyclase hydrolyzes ATP to cyclic AMP, and cyclic adenosine monophosphate is a secondary messenger to activate protein kinase A. Protein kinase A phosphorylates a site on the sarcoplasmic reticulum, which increases calcium release. So we see here that protein kinase A increases trigger calcium by phosphorylating the slow inward L-type calcium channels, and it also increases calcium release from the sarcoplasmic reticulum by phosphorylating ryanidine receptors. To summarize the second method we've discussed of regulation of inotropy, again we have epinephrine and norepinephrine, which will bind to their respective receptors. And these will be coupled to our G stimulatory protein. This will activate our adenyl cyclase, which will take ATP and convert it to cyclic AMP. AMP will activate our protein kinase A, and this time we'll follow protein kinase A towards our sarcoplasmic reticulum, which when our ryanidine receptors are stimulated on it, we will have increased calcium efflux from our sarcoplasmic reticulum, increasing the intracellular concentration
that can be utilized for binding our troponin C.
The minor pathway by which the sarcoplasmic reticulum is stimulated to release calcium is as follows. Sympathetic nerve release or adrenal release of norepinephrine into the circulation, or creation of angiotensin II by angiotensin converting enzyme, or endothelial release of endothelial 1 will create substrate that bind to specific receptors. Norepinephrine will bind to alpha-1 receptors, angiotensin II binds to AT1, angiotensin II receptors, and endothelial 1 binds to ETA, endothelial 1 receptors. Each of these receptors are coupled to GQ stimulatory proteins, and the GQ protein stimulates phospholipase C. Phospholipase C converts phosphatidylibositol, 4,5-biphosphate, or PIP2, to inositol triphosphate, or IP3. IP3 phosphorylates a site on the sarcoplasmic reticulum, stimulating it to increase calcium release.
Summarizing our minor pathway for sarcoplasmic reticulum release of calcium, modulation of inotropy. Again, if we have norepinephrine, angiotensin II, or endothelin, these are going to stimulate their respective receptors, and these receptors are going to be coupled with a GQ protein, and the GQ protein is going to activate our phospholipase C. Phospholipase C will take PIP2 and convert it to IP3, and IP3 is going to phosphorylate again our ranitidine receptor on our sarcoplasmic reticulum, such that we have increased calcium efflux into our intracellular space, once again available for troponin C to be bound to.
Calcium binding to troponin C. Calcium binds to troponin C, and this is determined by the degree of free intracellular calcium concentration, and the binding affinity in the presence of a specific concentration is also affected by acidosis, which has a negative effect on calcium affinity, and preload, where an increased preload will increase the affinity of troponin C for calcium. Myosin ATPase activity, or myosin phosphorylation. Increased cyclic AMP is associated with increased phosphorylation of myosin heads by myosin light chain kinase. The rate of association between myosin and ATP leads to increased strength of contraction. One way that beta-adrenergic stimulation increases inotropy is by increasing actin-myosin complexes by phosphorylation. Calcium uptake by the sarcoplasmic reticulum, or sarcoendoplasmic reticulum calcium ATPase activity. Increased calcium in the sarcoplasmic reticulum by SERCA uptake indirectly increases calcium release of sarcoplasmic reticulum stores. So enhanced sequestration of calcium by the sarcoplasmic reticulum at the end of one contraction increases the subsequent release of calcium and stimulation for the next contraction. 80% of intracellular calcium is eliminated by uptake into the sarcoplasmic reticulum. Protein kinase A phosphorylation of phospholambin decreases phospholambin's inhibitory effect on SERCA, thereby increasing the rate of calcium into the sarcoplasmic reticulum. SERCA activity is increased by intracellular calcium, which is caused by increased calcium entry into the cell and decreased calcium efflux. Hypoxia and the associated decreased APT production will affect SERCA because it requires APT for functioning. So there will be decreased calcium movement in and out of the sarcoplasmic reticulum in the deficit of ATP.
Summarizing our SERCA activity, we have norepinephrine and epinephrine, again these binding to their respective receptors, and the receptor will be coupled to, again, a G stimulatory protein, which will activate our adenyl cyclase. Adenyl cyclase will take ATP and change it to cyclic AMP, and again cyclic AMP is going to make our protein kinase A. And now normally in our sarcoplasmic reticulum, our SERCA pump, which is an ATP dependent pump that allows calcium to go into the sarcoplasmic reticulum at the end of contraction. Normally this is inhibited by phospholambin. protein kinase A is going to inhibit that inhibitory effect such that the circuit can work better. The release of calcium by your ryanodine receptors at the beginning of each contraction is indirectly influenced by the amount of uptake from the prior contraction. The greater or the better the circuit pump works, the better the next release and subsequent inotropy. So to summarize, inhibition of your circuit activity is going to come again from either acetylcholine or adenosine binding to their respective receptors, this being coupled with inhibitory G protein, which will have a downregulatory effect on adenyl cyclase. ATP will then not go to cyclic AMP. And the rest of the pathway won't take place. The inhibition of our phospholambin on circa will not be there, so you will have limited calcium uptake rather than if this pathway were to be stimulated into your sarcoplasmic reticulum. Meaning for your next calcium release, you would have a potentially lesser amount and less inotropy.
The last mechanism we'll be looking at is regulation of calcium myocyte efflux. Again, 80% of intracellular calcium is taken up by the sarcoplasmic reticulum. 20% of the remaining intracellular calcium is eliminated by membrane mechanisms, 15% by exchangers, and 5% by pumps. And the exchanger is a sodium-calcium exchanger, the inhibition of which will increase intracellular calcium levels. And the second is the ATP-dependent calcium pump.
And so to summarize, calcium efflux, or removal of calcium at the end of a contraction cycle, if you remember, 80% is going to go into your sarcoplasmic reticulum, which means we have 20% to leave the cell itself, and 15% will occur via an exchanger, and this is going to be a sodium-calcium exchanger, and 5% is going to occur via a calcium ATPase pump.
Methods of affecting these pumps, one would be to give a cardiac glycoside, such as digoxin. This would decrease sodium-potassium ATPase activity, causing a rise in intracellular sodium. This elevated intracellular sodium level would decrease sodium-calcium exchange activity, leading to a rise in intracellular calcium, and thereby increasing inotropy. Hypoxia and its resulting decrease in ATP will also decrease sodium-potassium ATPase pump activity and will bring about the resulting electrolyte changes, including a rise in intracellular calcium. However, inotropy is not increased in the presence of hypoxia because of the deficiency of ATP, which is required for myosin ATPase. The opposite of inotropy is lusitropy, the both of which, again, are energy-dependent processes. Myocyte relaxation is determined by the cell's ability to decrease intracellular calcium. The primary mechanism is calcium uptake into the sarcoplasmic reticulum by the circopump, again, 80%. Decreased intracellular calcium causes calcium that is bound to troponin C to release, so troponin tropomyosin complexes resume their inactivating position. Binding affinity of troponin C modulated by pKa phosphorylation of the troponin I leads to increased calcium dissociation from troponin C and increased relaxation. 4 of the mechanisms that contribute to lusitropy regulation are: number 1, decreased circuit activity by decreased phosphorylation of phospholambin, which will retain its inhibitory effect, leading to calcium overload intracellularly and impaired relaxation. Number 2, hypoxia causes increased calcium permeability, leading to increased calcium influx and calcium overload and impaired relaxation. Number 3, inhibition of sarcolemia calcium ATPase pumps or sodium-calcium exchangers, causing calcium overload and impaired relaxation. Number 4, binding affinity to troponin C modulated by decreased pKa activity phosphorylation of troponin I leads to decreased dissociation of calcium from troponin C and impaired relaxation. The fourth mechanism is unique in that it will increase inotropy, but it will decrease lusitropy. Diastolic dysfunction is when there is too much calcium inside of the cell during contraction and too little leaving after contraction, or too little taken up by the sarcoplasmic reticulum. If this concentration level remains high, calcium fails to leave its binding sites on troponin, and actin-myosin cross bridges do not disengage. Moving on to vasculature, we will look at the anatomical structure of your blood vessels. Moving from inward to outward, your blood vessels consist of an inner lumen, an intimal endothelium, your basal lamina, which plus or minus has connective tissue, which is only found in the larger vessels, the media and its circumferential and helical longitudinal muscular layers, the external elastic lamina, and the adventitia. And focusing on the three main layers, the intima, the media, and adventitia, and going into further detail, the intima is a single layer of epithelial cells. The media consists of smooth muscle cells, collagen, and elastin in a ratio that determines their vascular behavior. The number of smooth muscle layers is associated with vessel size. The aorta has the greatest proportion of elastin. This allows it to passively expand and contract and to dampen arterial pressure. The smaller arteries and arterioles have the greatest amounts of smooth muscle, and this allows them to contract and regulate arterial pressure and blood flow. The capillaries themselves do not have any smooth muscle and consist only of endothelial cells and a basal lamina. The adventitia contains collagen, fibroblasts, blood vessels, or your vasa vasorum, lymphatics, and autonomic nerves, primarily sympathetic adrenergic. The typical vascular smooth muscle is 5 to 10 nanometers in diameter and 50 to 300 nanometers in length. Similar to the T-tubules of the cardiac myocyte, vascular smooth muscle has surface caveolae. These as well serve the purpose of increasing surface area. They do have a sarcoplasmic reticulum, however, it is less developed than the cardiac myocyte. Actin and myosin, while still present, are not arranged in the distinct bands of repeating units associated with myocytes. Actin is joined together end to end by dense bodies within the cell and dense bands to the inner surface of the cell. Electrically, they are connected by gap junctions, which allow propagation of action potentials along the vessel. Vasculature smooth muscle contraction is slow and sustained. The normal resting state is actually partially contracted. This tonic contraction is determined by the stimulatory and inhibitory effects acting upon the vessel. Sympathetic adrenergic nerves are the most important and cause a certain amount of stimulation and contraction. Circulating catecholamines such as epinephrine and angiotensin II also play a role. Substances released by the endothelium we will see also have an effect on its tonic state, and substances released surrounding blood vessels. The initiation of contraction is brought about by three mechanisms: electrical stimuli, chemical stimuli, and mechanical stimuli. Electrical stimuli open voltage-gated slow inward L-type calcium channels. Changes in ion concentration or receptor coupling to calcium ion channels will affect this electrical stimulation as well. Chemical stimulation can bring about contraction through mediators such as epinephrine, norepinephrine, angiotensin II, vasopressin, endothelin-1, and thromboxane A2. All bind to their specific receptors and all eventually increase intracellular calcium. Mechanical stimuli can bring about contraction through passive stretching, and this is called a myogenic response, and stretch-induced activation of calcium channels. Smooth muscle contraction is brought about by the increase of intracellular calcium either by increased L-type calcium channel influx or increased sarcoplasmic reticulum release of stored calcium. Calcium binds to the protein calmodulin, and the calcium-calmodulin complex activates myosin light chain kinase. Myosin light chain kinase phosphorylates myosin light chains in the presence of ATP. This creates a cross-bridging between myosin heads and actin filaments and subsequent smooth muscle contraction.
To summarize smooth muscle contraction, again we have calcium entering the cell through our slow inward L-type calcium channels, acting as trigger calcium for our sarcoplasmic reticulum, and again calcium is released from our sarcoplasmic reticulum. In the presence of calmodulin, calcium forms a calcium calmodulin complex, and this will activate our myosin light chain kinase. Myosin light chain kinase is an enzyme that allows for hydrolyzation of our myosin light chain in the presence of ATP. And this will change the conformational position of the globular head and energize it and allow it to bind and have potential energy for contraction when the actin-myosin cross-bridge formation is completed.
Smooth muscle relaxation, calcium is resequestered by ATP-dependent calcium circle-like pumps on the sarcoplasmic reticulum, and calcium is removed from the cell by ATP-dependent calcium pumps or sodium-calcium exchangers. So the question is, how do we affect these mechanisms to bring about smooth muscle contraction or relaxation? And there are three basic pathways which can modulate smooth muscle activity. The IP3 pathway will bring about contraction. And that is the IP3 via the GQ protein activation of phospholipase. Relaxation can be brought about by cyclic AMP or cyclic GMP. Cyclic AMP via the GS protein activation of adenocyclase, and cyclic GMP or cyclic guanosine monophosphate via nitric oxide activation of guanylyl cyclase. Let's look in depth at each of these pathways. The first brings about contraction. It is the IP3 pathway. Epinephrine, norepinephrine, angiotensin II, endothelium 1, vasopressin, acetylcholine, all of these bind to their receptors respectively, alpha 1, AT1, ETA, vasopressin 1, and M3 receptors. Each of these receptors is bind to GQ proteins. These GQ proteins activate phospholipase C. PIP2 phosphorylates to IP3 by PLC, and IP3 stimulates sarcoplasmic release of calcium.
To summarize the major pathway of the IP3 pathway of contraction of vascular smooth muscle, if we have norepinephrine, angiotensin II, endothelin, acetylcholine, or vasopressin, all of these will bind to their respective receptors. This is going to stimulate a GQ protein, which in the presence of GTP, going to GDP and inorganic phosphate, we will find that this is going to make our protein lipase C, and protein lipase C will allow for PIP2 to phosphorylate to IP3. IP3 will stimulate our sarcoplasmic reticulum to again release calcium and thus bring about contraction.
There's also an alternate branch pathway through which IP3 operates. The same list of molecules attached to the respective receptors as in the last description, and again are coupled to the GQ protein, and the GQ protein activates phospholipase C. Phospholipase C causes PIP phosphorylation to IP3, and as a byproduct of formation of DAG, diacylglycerol. DAG activates PKC, and PKC modulates smooth muscle contraction via phosphorylation. The cyclic AMP pathway brings about relaxation. Here, epinephrine or isoproterenol bind to beta-2 adrenoreceptors, and adenosine or prostacycline or PGI2 bind to their own respective receptors. Each of these receptors is coupled to a GS stimulatory protein, and this GA protein stimulates adenocyclase. This increases cyclic AMP, which inhibits myosin light chain kinase, thus inhibiting myosin light chain phosphorylation, thus inhibiting cross-bridging between myosin heads and the actin filaments. In this way, contraction is inhibited and muscle relaxation is brought about.
Summarizing smooth muscle relaxation via the cyclic AMP pathway, epinephrine, adenosine, or prostacycline, again, each are going to bind to their respective receptors. These will be coupled with a G stimulatory protein, and in the presence of GTP going to GDP and inorganic phosphate, these will stimulate our adenyl cyclase. And our adenyl cyclase, once again, will say ATP going to cyclic AMP. And cyclic AMP is going to downregulate myosin light chain kinase. Myosin light chain kinase has an enzyme that allows for phosphorylation in the presence of ATP going to ADP and inorganic phosphate of your myosin light chains on the myosin heads. So if this is inhibited, we do not have phosphorylation of our myosin heads, and you have decreased myosin actin cross-bridging formation and decreased contraction, and thus smooth vessel relaxation.
The nitric oxide cyclic GMP system, here acetylcholine and bradykinin and substance P, when bound to receptors, convert L-arginine to nitric oxide in the presence of nitric oxide synthase. Nitric oxide diffuses relatively easily from the endothelial cells into vascular smooth muscle. Nitric oxide activates guanylyl cyclase, which increases cyclic GMP formation and resulting smooth muscle relaxation. Cyclic GMP also decreases calcium entry into vascular smooth muscle by inhibiting slow inward L-type calcium channels. It activates potassium channels, causing cellular hyperpolarization, and decreases the IP3 pathway channel activity, thus again bringing about smooth muscle relaxation. The vascular endothelial cells also become important to discuss. The endothelial cells are flat, single-nucleated, elongated cells. Intracellular junctions differ by vessel type and tissue location. They are tight in the arteries and in skeletal muscle, and they are very wide in the capillaries of the spleen and bone marrow. And the endothelial cells have several functions. They function as a barrier for exchange of fluid, electrolytes, macromolecules, and cells between the intravascular and extravascular space. They regulate smooth muscle function by nitric oxide, prostacyclin, and endothelin. They modulate platelet aggregation by biosynthesis of nitric oxide and prostacyclin, and they modulate leukocyte adhesion and transendothelial migration through biosynthesis of nitric oxide and expression of surface adhesion molecules. When vascular endothelial cells secrete endothelin-1, this brings about contraction. Endothelin-1 binds to ETA on vascular smooth muscle and activates the IP3 pathway. This causes calcium mobilization, smooth muscle contraction, and vasoconstriction. It can be stimulated by angiotensin II, vasopressin, thrombin, cytokines, and shearing forces on the vessel. It is inhibited by nitric oxide and prostacyclin, which have negative effects on smooth muscle contraction. Prostacyclin also brings about relaxation. Prostacyclin is a product of arachidonic acid metabolism in endothelial cells. It exerts its effects through the cyclic AMP pathway, thus causing smooth muscle relaxation, and it also causes inhibition of platelet aggregation. Nitric oxide as well brings about smooth muscle relaxation. Endothelial cells continuously produce nitric oxide. Production is enhanced by acetylcholine and bradykinin, which are agonists binding to epithelial receptors, increased shearing forces on the endothelial surface, and cytokines such as tumor necrosis factor or interleukins, which are released from leukocytes. Endothelial-derived nitric oxide works through increased activation of cyclic GMP. This relaxes smooth muscle, and it also inhibits platelet function and aggregation, and it inhibits inflammation. When there's endothelial damage, be the source atherosclerosis, hypertension, diabetes, or hyperlipidemia, this causes decreased nitric oxide and prostacyclin production. This leads to chronic vasoconstriction, less vasodilatory capacity, thrombosis, and vascular inflammation. It also leads to increased capillary permeability and fluid infiltration and edema. Increased endothelin-1 production can lead to hypertension and vascular damage and the above process.
Let's take everything we've learned up until this point and put it together in a clinical context. We'll talk about a drug. Specifically, we'll talk about milrinone. And milrinone, if you remember from pharmacology, is a PDI, a phosphodiesterase inhibitor. Well, a phosphodiesterase is an enzyme that degrades cyclic AMP. So we know it is going to affect the cyclic AMP pathways of your myocytes, and it is going to affect the cyclic AMP pathway of your vascular smooth muscle. And so let's take a look at each and what happens when this is inhibited and what the end result is. So again, norepinephrine and epinephrine are going to stimulate our beta-1 receptors, coupled with G-stimulatory proteins are going to stimulate adenylene cyclase, and this is going to take ATP and convert it to cyclic AMP. This will promote protein kinase A, and protein kinase A has several effects. It phosphorylates our slow inward L-type calcium channels such that we have increased trigger calcium. It stimulates our ryanodine receptors on our Sarcoplasmic Reticulum such that we have greater calcium efflux into the cytoplasm available for troponin C. And it also inhibits the inhibitory effect of phospholambin on our Circa ATP pump, allowing for greater calcium uptake from the previous contraction cycle so that there's more calcium available to efflux from the Sarcoplasmic Reticulum for this cycle. So in 3 ways, cyclic AMP increases our inotropy. Well, if I have a phosphodiesterase and this is inhibiting that cyclic AMP and its downstream pKa, what you're going to find is if I have a phosphodiesterase inhibitor, it will inhibit this inhibition. All right? And by taking away that inhibition, you have greater cyclic AMP and protein kinase A and the resulting positive inotropic effects. So milrinone is a positive inotrope. Okay? In our vascular smooth muscle, again, we have epinephrine, adenosine, prostacyclin, each stimulating its own receptor. As well, this will be grouped with a G-stimulatory protein, and GTP is going to go to GDP, and inorganic phosphate will have a positive effect on amylyl cyclase. ATP will go to cyclic AMP, and cyclic AMP has an inhibitory effect on myosin light chain kinase. Myosin light chain kinase is the enzyme that phosphorylates myosin light chain on the head of your myosin, and in the presence of ATP going to ADP and inorganic phosphate. So what we find here is that cyclic AMP inhibits this enzyme and thus inhibits phosphorylation of your myosin head and myosin-actin cross-bridging and contraction. So if I have a phosphodiesterase, which is going to inhibit that cyclic AMP, that means that inhibitory effect is taken away. I have more myosin light chain kinase available to phosphorylate my myosin light chains and greater actin-myosin bridging and cross-bridging. If I have a phosphodiesterase inhibitor, the inhibition is inhibited. So I now have unopposed cyclic AMP, which will inhibit my myosin light chain kinase. And therefore I will have less phosphorylation of my myosin light chains and less actin-myosin cross-bridging. And therefore I will have relaxation, vasodilation, and in that way you have relaxation of your vascular smooth muscle. So you have positive inotropy and you have vasodilation. And those are the characteristics of milrinone.
Taking the functional microanatomy discussed at the beginning of the lecture and the molecular physiology discussed at the second half of the lecture, we once again have to zoom out and develop a new understanding. And the springboard will be our length-tension relationship. The length-tension relationship is the concept that the force generated by the contraction of the sarcomere is related to the degree of myosin and actin overlap. If the sarcomere is overshortened, thick filaments can run into the Z line and become disordered. Thin filaments can cross the M line and interact with each other or with myosin cross bridges from the other half of the sarcomere. In the example of a hypertrophic cardiomyopathy, Filling pressures are less likely to stretch muscle at its resting cell length. With a dilated cardiomyopathy, the sarcomeres are overstretched with the resting diastole, which causes decreased pressure building capacity within the ventricular muscle. The microanatomy and molecular physiology discussed come to a culmination on this slide, which is the basis for our understanding of our Starling curve. This is a good place to take a break. Once again, we've ended by zooming out and moving away from looking at one cell to looking at a tissue as it functions together. We've covered all of our relevant microanatomy of both our myocytes and the smooth muscle within our vasculature, and gone over the relevant molecular physiologic processes such that we have a firm ground to stand on when we move on to more complex tissue-based physiologic lectures. Okay, once again, this looks like a good place to take a break. I hope you found the lecture both enjoyable and beneficial. I'd say that one of the truly great things about being a surgical cardiothoracic physician assistant is the many hats that you get to wear in the pursuit of proficiency within the field. Not only are you a clinician, but you're a surgeon and a pharmacist, and in some ways you're an electrician of the heart and a plumber of the heart. And I hope you've recognized that the first lecture focused on that electrician profession. And going back to that first lecture, it serves as a good springboard towards moving up towards our conduction system and looking at issues of pharmacology and manipulating dysrhythmias of the heart and EKGs. And this lecture is a good springboard towards the plumber profession of the heart and looking at the hemodynamics and the muscular structure and function relationships of the heart. So whichever you choose is up to you. Eventually you'll cover them all, but your interest will guide you. And I hope that this lecture has been interesting for you, and I hope that you come back for the next one as well. As always, thank you for your time and your interest.